All right, hello. I'm Brianna Barrett. I'm very pleased to be here at Meeting on the Med for this presentation, and we're excited to share with you today some really promising and exciting innovations coming out of Forge Biologics. Maybe. Oh, there we go. All right, so here is a little bit about Forge to begin. So we're a leading global genetic medicine CDMO. We're currently focused on AAV technologies. We have about 20 GMP suites currently operational. These have bioreactors scaled from 1 to 5,000 liters. This can process about 20,000 liters of bioreactor capacity simultaneously, and we're currently a little bit over 370 employees strong. And this is all coming out of a very growing biotech hub in Columbus, Ohio. So we currently have over 50 clients that we're serving, all in the gene therapy space. Most of these clients are using our platform technologies. So we have 93% of our clients utilizing our Ignition HEC-293 cell line, and 84% utilizing our PMBR Ad Helper plasmid. The therapeutic areas that our clients are targeting are pretty vast, spanning from the ocular space all the way through musculoskeletal diseases. The serotypes to support these indications are vast as well. So you see experience in our platform process from everything from AAV9 all the way through AAV1, and also including novel serotypes. On the bottom, there's a list of some of our publicly announced clients. We're very proud to say that we're growing and developing into long-term partnerships. So some of the tools that we leverage to support the industry's evolving needs, we have a molecular improvement. So we have molecular development team that's constantly modifying our plasmids that are input into our suspension HEC-293 process. So this is really the initial goal of having enhanced productivity. We also have a process development team that's focused on process improvements, helping clients understand their critical process parameters, again, at achieving higher yields and also focusing on product quality. For our analytic development group and analytic validation group, they're really helping our clients focus on those critical quality attributes so they can understand their product and their process. And here we're staying at the forefront of analytical technologies that are available in the field and constantly striving to bring more and more resources in-house. And then, of course, there's the traditional scale up and scale out, your most efficient path forward. So I wanted to provide a brief comparison of the 4 major systems for producing AAV. So we have the traditional transient transfection HEC-293 system, baculovirus SF9, herpes virus HEK-293, and then also producer cell systems. Each of these systems have pros and cons. For the transient transfection method, which is utilized at Forge and is utilized by many of our partners in the field, what's plagued this method is it's on the lower end of yields while it's on the higher end of cost of goods. This is known, and this is what we strive to constantly improve. So what I'll share here today is kind of a change in the narrative where Forge is now producing batches using a suspension platform method that have increased yields drastically while then simultaneously decreasing the cost of goods per dose. All right, so why are we focused on manufacturing innovation? We have a platform process. We could routinely go through the motions of performing this process. So most of the senior leadership and many of the members of Forge started out as gene therapy developers, including myself. So we recognize that as disease indications, we reach for more prevalent indications, and our clients are bringing more and more programs. Manufacturers must keep pace, and we feel that responsibility as the CDMO. So manufacturing innovation and R&D specifically have been a key part of the Forge and Ajinomoto collaboration. And we're very grateful to have partners with the same beliefs as ourselves. So late last year, we launched our fuel platform process. And the goal of this was to deliver more efficient manufacturing solutions so we can support more programs and larger diseases, all supporting our company mission as well to enable access to gene therapies. So I've mentioned fuel a lot. What is the fuel platform? So the fuel platform is really a combination of our advanced technologies, some molecular improvements, in combination with optimization packages, and then all built upon our platform process. So the two advanced technologies that I'll really touch on today are our PMBR 2.0 Ad Helper plasmid, and then also our modified RepCap plasmid. Okay, so at the top here, we have our PMBR Ad Helper plasmid. This is one of the smallest available, commercially available ad helpers at 8.9 kb. This not only increases the efficiency of manufacturing the plasmid itself, but it also increases the efficiency of the AAV that it's manufacturing, and we'll show some data shortly. Some of the elements that have been removed or reduced in this plasmid are shown here, and it's all with the goal of increasing and improving the safety profile. Next, we have our modified RepCap plasmid. So for this technology is really a backbone that we're providing, and we're able to take a client's capsid sequence or your original RepCap worked on at your company, whether it is novel or a natural serotype, and we're able to insert that into our backbone with no protein sequences, no protein sequence changes, excuse me. And again, this is for safety improvements and higher AAV yields. And then, of course, these are put into our manufacturing system in combination with our ignition cells. So here's some of the data coming out of this program. So we're seeing, what you're looking at here is a graph across serotypes, and you're looking at clarified lysate productivity, so what's coming out of the upstream process. What we're seeing here is with only the input of those two novel technologies, we're seeing increases of 12 to 15%, or I guess as low as 3x fold, not percent, excuse me. So we're seeing 3 to 15 fold over industry standard titers. Where this has been most impactful is for those traditionally lower or harder to produce serotypes, such as AAV2. And you can see from this graph, we're now achieving with AAV2 what we used to really only see productivity-wise out of AAV9. Next, we wanted to see, have we really maxed out our productivity with these technologies? Is the system, fully saturated, or is there further improvement that can be made? So next, we took those two molecular improvements and tried to optimize per the product we were making. So we did this with the transfection DOE. It's probably the most critical unit operation for upstream yield improvements. Here you see the orange bar is the fuel platform with the plasmid input alone. The blue bar is the plasmid input with the transfection DOE. And we're seeing then a further increase. So now we're getting 13 to 22 times what was previously seen in upstream productions. Okay, there we go. All right, so next, this is my absolute favorite slide. We're showing not only the scalability, but some downstream critical quality attributes. So it's not just yield, we're also maintaining quality with this platform. So the orange bar here, you're still looking at productivity, so your viral genomes per liter. Your orange bar is a shake flask. The red bar is a 200-liter single-use bioreactor. We've since taken this to 500 and 1,000-liter bioreactors as well, and are seeing enormous productivity gains. For critical quality attributes, we've looked at empty-full ratio, so we still get about 90% full capsids after cesium enrichment. We have purity profiles of under 1 EU per mil for endotoxin, and our capsid purity is still around 99%, so this is excellent. And then the best piece, or to me, the most impactful piece of data here is actually your recovery. So a typical recovery in the field is maybe around 20 to 30% across all the purification steps of the process. And this would be typical. What we're seeing here is that we have right-sized the process. Typically, equipment and resins and columns are oversized to allow processing within a reasonable duration, say 24 hours for a unit operation. Now we have enough viral genomes per volume that we're really capturing everything. What you're making, you are keeping, and we're seeing about 70% recovery across the process. All right, so as I mentioned, Forge has built the fuel process really on the basis of our platform process. So we've completed to date over 500 batches of AAV, so mostly at the 1,000-liter scale, but also ample experience at the 550-liter scales. To recap on the fuel process, our advanced technologies are leading to 2 to 15 times improvement in productivity over industry standard titers. And these technologies are designed to be safer. In combination with optimization packages with studies such as a transfection design of experiment, we're seeing further increases up to 22 times over industry standard. And this is all with comparable product quality attributes. And then again, this is all built upon our original platform, so we're able to leverage those years of data and experience that we've gathered. So on the right here, you see the impact of all of this. So what was previously requiring a 1,000-liter bioreactor now can be accomplished in a 50-liter bioreactor for about a 20-time efficiency. All right, so I'll end the talk today with a bit on how we collaborate with our European partners as we're here at the beautiful meeting on the Med. So we are currently supporting multiple clients with clinical trials in the EU. So this is a routine experience for us. What we are able to provide is our facility currently meets all EMA classification requirements. We have a full suite of packaging and shipping offerings for a clear line of sight into global supply chain. We also have had a quality person audit our facility, and we're granted QP declaration for both drug substance and drug product processes. Again, to support clinical programs, we've had multiple clients go through the QP release process on their batches as well successfully. And we are very proud of our integrated global regulatory services, where our regulatory team has supported multiple clients in completing CTA. All right, and with that, thank you very much. If anyone has any questions, please feel free to contact me or any of my colleagues. We'll be hanging out towards the back, but thank you.